All right, so hello again. I'm Martin Kampmann, and I was a postdoc in Jonathan Weissman's lab until about a year ago when I started my independent lab at UCSF. And what I will talk to you about today and also tomorrow are multiplex CRISPR screens. So we have today until about noon. Today I actually have to leave relatively soon afternoon as well, but I will be there tomorrow for the whole afternoon workshop session where we'll actually apply some of the bioinformatics techniques to analyze these kinds of screens together. And there will be plenty of opportunities for you to ask me questions then. We'll see how far we get today. We also have a bioinformatics lecture session tomorrow with Jacob Korn. And today I'm mostly going to focus on the why and the big picture, how, why, what. And then we get into more of the technical details tomorrow. So I think one thing that's probably in common to all of us here is that we're curious about how genes determine biology and disease. And this is a slide that I show often when I talk to a cancer audience. And I think in this audience, I'm going to preach to the choir when I'm contrasting two fundamentally different approaches to understanding these connections between genes and biological processes. We can take an observational approach, looking at human genetics and a lot of other genomics, omics tools where we can observe mutations, we can observe gene expression patterns, and correlate that with different phenotypes, diseases, developmental stages, and so on. The approach that we'll be talking about today is a functional genomics approach where we perturb the system, we perturb gene function and then observe the functional consequences. So intrinsically, these observational omics approaches are based on correlation versus in a system that enables us to perturb gene functions can really lead us to understand causal relationships. Also, importantly, we have to remember that when we want to understand, for example, in the context of a cancer cells, what are the functional consequences of all genes in those cells, and we do that based on observing naturally occurring patient samples, we're really underdetermined. We're not going to be able to recapitulate all the possible mutations and all their combinations. Versus in a functional genomics experiment, we can actually design a controlled experiment in an isogenic background. Vice versa, obviously, if we're observing, say, cancers in human patients, we're actually working with the real thing. Our functional genomics approaches are always going to happen in model systems. So ideally, we want to really integrate these two types of approaches. But as I said, today we're going to talk about how we can use CRISPR for this functional genomics approach. And we heard already several examples about that from Dana and from Luke. I think one important distinction that I want to introduce at this point is this distinction between forward genetics and reverse genetics. It's actually a little controversial how we define these things. Clearly, I think a lot of the examples that we've heard about so far were reverse genetics, where you start from a gene of interest and you characterize its phenotype by perturbing that gene. And so my definition for these two types of genetics is reverse genetics means you're starting from a gene of interest and look at a phenotype. Forward genetics means you're starting from a phenotype of interest and ask what are all the genes in the genome that are relevant, that control or contribute to this phenotype. And what that means is that whereas in reverse genetics we can perturb one gene at a time, for forward genetics approach we need to have a very scalable system to perturb basically all genes and read out their impact on the phenotype we care about. And so this is where these multiplex CRISPR screens that are the topic we want to talk about today come in. So what are different ways to manipulate gene functions in human cells? The first category of such approaches are approaches that alter the actual genome. And obviously, people have tried to do this for a long time in various organisms. I say human cells here because a lot of the work that I will be showing you is in human cells in culture. It might as well apply to any other mammalian cells or many other cell systems. What I'm not going to talk to you about today are whole organism screens, and we'll hear from other people more about applications in whole organisms. So today what I talk about mostly are applies to unicellular organisms and isolated cells in culture. So people have used random mutagenesis approaches before, using, for example, chemical mutagens, as well as, I'll talk a little bit more about that, and we had a beautiful poster last night with a really good application of this technology, using insertional transposon mutagenesis to randomly disrupt gene function in the genome. In addition to these random mutagenesis approaches, there can be site-directed mutagenesis and editing approaches, homologous recombination using site-directed recombinases, and obviously, as we heard from Dana yesterday, combining cutting with either homologous recombination or non-homologous end joining approaches, which traditionally or earlier could be done with zinc finger and talons, and now can be done with CRISPR-Cas9. We can go into the pros and cons of using these approaches in a minute, but I think some people think only of random mutagenesis approaches as truly forward genetics, because these enable you to really, if you have the capacity to screen a lot of genotypes, to really discover completely new things. If we take approaches that are already focused on, say, an annotation of a genome and on certain genes, some people consider that to be a reverse genetic approach. But if we do this at genome scale, I think we're approximating a true forward genetic screen with this. And as I will show you, it has many practical advantages. So this slide has ways to perturb genes by altering the genome. As we heard about extensively from Luke yesterday and today, we can also manipulate gene expression and thereby perturb gene function. And we can manipulate the transcription by activating or repressing endogenous promoters using, again, TAL fusions or CRISPR-IA approaches. We can express transgenes, for example, by introducing a cDNA into a cell. So that is manipulating gene transcription. We can also manipulate the stability of transcripts, for example, using RNAi interference and newer CRISPR-based technologies that also act on RNA. We can also control the stability of protein products themselves by introducing degrons that could be controlled, for example, by small molecules. And lastly, we can also manipulate gene product functions of pre-existing gene products by using pharmacological tools. So I think these are the main categories that we can experimentally manipulate gene functions. And as you'll see, some of them are great for a one-off experiment, but not all of them really lend themselves to a genome-wide approach where we want to do this in a comprehensive genome-wide fashion. One method that I'm just going to mention that is from the pre-CRISPR era, but that still has its uses and, as I said, has some beautiful applications, is insertion mutagenesis, by which, for example, a mammalian cell line is infected with in a pool with a let's call it here gene trap virus, basically an element that inserts randomly into the genome, disrupts the genes where it's integrated with a known sequence, and thereby enables you to select cells with a given phenotype and identify, using sequencing, the insertion site. The main challenge is that human cells are naturally diploid, and so this random disabling of genes will miss out a lot of genes if you have a recessive phenotype, unless you would do it in a haploid cell line. So one of the big innovations for this method was to use a haploid cell line, and Tyne Brommelkamp has really pioneered this. As I said, it can give you good results, but in this implementation, it is really limited to these very specific haploid cell lines, and you couldn't do it in your cell line of interest, such as, say, a patient-derived cell line or as another specific cell type. The other method that clearly has been used a lot to manipulate gene functions in human cells, again in the pre-CRISPR area, was RNA interference. And RNA interference relies on an endogenous system that is used for host defense and also gene regulation, whereby basically double-stranded RNA molecules are processed by an endogenous enzyme called dicer to form short interfering RNAs. Oh, I should use a pointer. Short interfering RNAs, and those can act in two ways. They can base pair with messenger RNA molecules and direct degradation of the messenger RNA. So this is a way to reduce the transcript level. And this requires a pretty high degree of complementarity, and that's where specificity in the system comes from. Unfortunately, for the purposes of using this as a precise tool, it also acts through a second pathway that is generally not directing transcript degradation, but translational repression of the target. And this pathway only requires a very limited amount of base pairing complementarity between this short RNA molecule and the target. Because this is such a limited amount of complementarity that is needed for translational repression, it's actually almost impossible to design specific reagents that would not have off-target effects on other genes in the genome. It's very hard to predict them as well because it's such a degenerate specificity. Nevertheless, the system has been used, but it's also gotten a bad reputation. This is maybe the most infamous example, and this is a meta-analysis of different HIV host factor screens that have been done. Clearly, this was an area of great interest, great medical interest. So different labs did RNAi screens to identify human cell factors that would be required for HIV infection, HIV replication. And as you can see, four of these screens that were done, all 2008, 2009, if the genes were overlapped, basically there were no genes that were detected by all four screens, and the overlap is essentially as much or less than you would expect by chance almost. So it's pretty poor and definitely turned people off using RNAi for a while. Having said that, the same paper actually also showed that, say, proteomics methodologies also had a poor overlap. So partially, we might say some of this has to do probably with the fact that different labs use slightly different cell lines, slightly different ways to set up the assay and get very, very different answers. But the idea then was that we have several things conspiring here. We know that siRNAs have very strong off-target effects that essentially we can't overcome, right? And this can lead to false positive results. Also, not all of them will be active, so that can contribute to false negative results. And obviously, any experiments you set up has measurement noise that contributes to both of these false results. And that turns into all these factors come together to make it a challenge to conduct good screens. So one of the things that I helped to develop in the first half of my postdoc in the Weizmann lab was to try to overcome some of these issues with RNAi by saying, well, maybe some of this is just a numbers game. What if we target each gene in the human genome by a very large number of independent reagent, in this case shRNAs, and we also have a lot of negative control shRNAs. What that will enable us to do is to really use a rigorous statistical framework to ask how likely is it that these 25 shRNAs designed to target gene A, any observed phenotypes here are more than you would expect it if you say pick 25 random shRNAs from your negative controls. And even though some of them are inactive, some of them have off-target effects, you can really do the statistics and ask, how likely was it that if this was not due to the real target of gene A, you would get such an accumulation of phenotypes for those 25 shRNAs. And the truth is that gave us actually highly robust results. And I would say that these complex libraries to this day are definitely among the best tools that we have and definitely on par with any CRISPR-based system at this point. The big drawback is, as you can see visually here, these are huge libraries, and this can be really prohibitive if you wanted to apply them to certain difficult systems. How did we do it? one of the first questions with these screens is, do you do an arrayed screen? Which means, do you have a way to distribute different reagents perturbing a given gene all individually into, say, wells, and then monitor cell phenotypes well by well? Well, for this library, a genome-wide screen would have taken more than 5,096 well plates. It's not impossible. Companies that screen drugs definitely operate at that scale, but it's pretty prohibitive, again, for a normal lab in academia. Instead, what we were able to do is to conduct a pooled screen, and I define that in a second, but that enabled us to conduct a whole genome-wide screen at this high coverage in a single spinner flask. So we can set up a spinner flask for, say, a suspension cell line, and then have a library of that complexity in a single flask, which is what every lab that does tissue culture can do. And what were the technologies that really enabled us to conduct these pooled screens? And other labs have done screens like that as well, obviously, at the time as well. For example, Steve Elledge's lab. The two technologies were the ability to synthesize high complexity oligonucleotide libraries, and we worked with Agilent on these libraries. So this is essentially using inkjet technology. Instead of printing 4 colors, it's printing 4 nucleotides and creating these slides that are also used for microarrays. In our case, we don't need them on slides. They're cleaved off, and we just have a tube of a highly complex set of oligonucleotides, and that encodes, all of the, say, shRNA, or as I show you later, sgRNAs that we want to have in our library. So we can package them into lentivirus, introduce them into cells, monitor phenotypes, separate cells by phenotype, and then we need to be able to ask which of these reagents had which effects. And their next generation sequencing clearly was a huge factor. And as you can see, this is actually taken from an NIH website that shows you how on an exponential scale, the cost of DNA sequencing has really bottomed out. This white line is kind of Moore's law, which describes how, in computer technology, the cost or the performance of processes improves, but in DNA sequencing has tracked that for a while. But then with next generation sequencing, it even dropped much faster. And this was, around the time that we set up this platform. So what's the principle then of how we actually practically implement these pooled screens? The basic principle is going to be the same, whether it's an shRNA screen, a CRISPR cutting screen, a CRISPR IA screen. Essentially, we're starting from a population of cells. They could be mammalian cells. They could also be microorganisms. In this case, the specifics apply to mammalian cells. We introduce a pooled library where we can clone these oligos into a lentiviral expression vector, for example, and then make a pooled lentiviral infection at a multiplicity of infections such that each cell, on average, will only be infected by one reagent. So this is important because we don't want to convolute effects of having different genes knocked down in a given cell at the same time. So we have this pooled cell population with different genotypes where essentially we integrate the expression cassette for an shRNA or sgRNA into the genome, and that enables us to stably manipulate gene expression and at the same time watermark that cell, barcode that cell, so we can always track it and know which guide RNAs was expressed because it's stably integrated into the genome. We can then select cells for a certain phenotype of interest, and we'll go through different use cases and different biological questions in a second. But one simple case would be, for example, to grow these cells just in the absence of a treatment, or to grow them in parallel in the presence of, say, a selective pressure using a drug, a toxin, and after several passages asking how did the frequencies of cells expressing a given guide RNA change during the experiment. And you could imagine here in this case, this green guide RNA knocking down gene B made the cells more resistant to the treatment, so it enriches in a population. So it's playing out Darwinian evolution in a tissue culture flask, if you will. And at the end of the experiment, we can harvest the cells, extract genomic DNA, PCR amplify the locus in the genomic DNA encoding the guide RNA, which is flanked by constant sequences that we know, and then quantify very precisely the frequency of cells that carry a different shRNA or guide RNA in these populations. what does that raw data look like? And we'll have you guys will get your hands onto actual raw data tomorrow and go through all the data analysis part in the workshop. But just to give you an idea, this is from one published study. The biology doesn't matter for now, but this is some raw sequencing data where on the x-axis we had cells from an untreated population. On the y-axis, a treated population, in this case, cancer cells treated with a drug that is used in the clinic. Each dot corresponds to one shRNA, in this case, in the population. And this is on a log2 scale axis, the number of sequencing counts we found for this shRNA. So in gray, you see all the negative control shRNAs. And to first approximation, these lie along a diagonal. The fact that they're not perfectly on one line reflects a combination of stochastic noise, which you're always going to have in a counting experiment like this, as well as probably true off-target effects that, especially in the case of shRNAs, are something we cannot avoid in principle. But what you can then do is you can then go through your shRNAs that are designed to target a given gene, one gene at a time. And in this case, this was our big library. So 25 dots here that are on this heat map that are overlaid on the negative controls. And what you can say is that these 25 shRNAs targeting PSMB5, some of them lie within this region of negative controls, but quite a few of them are very well separated. And then you can ask, again, given you know the distribution of negative controls, how likely was it to get such an arrangement just by chance and put a p-value on it? And in this case, it was very, very unlikely. 1 in 10 to the minus 10 times you would expect to find such an extreme distribution if these were just drawn from the negative controls. And similarly, we can detect genes that have the opposite effect. In this case, knockdown of PSMB5 made cells more sensitive to the treatment. Knockdown of this other gene, PSMB12, strongly made them resistant to the treatment. And so we can detect genes that have different effects like this. So this was just to set the stage of what we were able to do in the pre-CRISPR era. But a lot of the principles apply, and a lot of what I've shown you now is actually becoming simpler, more compact, and more straightforward due to CRISPR. Not long after CRISPR-Cas9 was first used in mammalian cells, two papers, one from the Sabatini lab and one from George Church's lab, and this was Sabatini and Landa as well, I think. Yeah, applied CRISPR-Cas9 nuclease screening. The library that they first proposed here in this Shalem et al. Paper, the Gecko library, is publicly available. There's a newer version of it now as well, and essentially it's the same principle. They synthesized an oligoarray encoding guide RNAs for cutting genes, and they introduced it in cell lines. And in this case, they grew the cells and compared between, say, day 3 and day 14, two different cell lines, what guide RNAs dropped out of the population. And they showed that the guide RNAs dropping out of the population targeted genes that were really enriched for essential processes, such as ribosome components here, things involved in RNA processing. And so, at least at this bird's eye view level, this method seemed to work very well. And since then, several papers have come out in different areas of biology that have successfully applied this technology. What does it rely on? As you guys remember from Dana's talk yesterday, just introducing cuts can have different outcomes. It can lead to a perfect repair, in which case the genomic DNA can be recut by the guide RNA. However, if there is a repair event that results in a frame shift, you can imagine that will really inactivate the whole gene and thereby lead to a loss of function that can have phenotypic consequences. Now, obviously, one thing that can also happen is that there is a repair event that, say, would result in a deletion of 3 nucleotides, in which case you would not get a frame shift event, you'd only delete 1 amino acid. And then that might not necessarily inactivate the gene. And a recent paper that I really like from Chris Vaccock's lab at Cold Spring Harbor Labs showed this very nicely. What they did is they targeted the lab is interested in chromatin modifying enzymes that are often very large proteins. And here you see in A and B two examples, BRD4 and SMARCA4. And this cartoon shows how along the length of this long proteins, there are different enzymatic domains and highly conserved domains, like an ATPase domain and so on. And what they did is they really evaluated a large number of guide RNAs that targeted several places along this long coding sequence for CRISPR-Cas9 cutting. Most of the published libraries all target towards the very 5' end. The idea being that if you want to get a frameshift and activate the gene, the earlier you make that frameshift, the better. But what they found is that the fact that not all cut events lead to frameshifts really obscured the strong negative dropout phenotype that he expected from these genes, and he knew to expect given his earlier RNAi work. And the strongest phenotypes he really picked up were just inside these important conserved and catalytic domains. So that's an important caveat. It's also an opportunity. The caveat is if you're doing screens with some of the available libraries, especially the ones that are really focusing on the 5' end of genes for cutting, one thing that can happen is the fact that not all of the editing events will lead to a frameshift. In some sense, if you think that the indel lengths are somewhat evenly distributed, and there's some quantitative papers that suggest that, you have for a single allele only a 2 in 3 chance to get a frameshift. Now, if you have a diploid cells, it's only 4 in 9 that both will have a frameshift. So that's less than 50% of your cases will have a clean frameshift in both alleles. And if your phenotype is a dropout phenotype, that could really obscure it in a genome-wide screen. So that's a caveat. And I know that the VACOG lab is making these libraries and making them available that are more focused on some of these domains or that blanket genes. So that's an interesting alternative approach. But the opportunity here is that obviously if you knew nothing about this gene and you would really blanket it with guide RNAs, you could really pick up patterns of where the important genes, functionally important regions in that gene are. And so you can actually learn additional information using this kind of analysis. This is assuming that loss of function of the gene does have a phenotype you can screen in this pooled screen. So that is one caveat. The other caveat is something that was reported that was obvious pretty early on, and right now, so first published as part of a paper in 2015, but 2 other labs have now in press papers that really have investigated more systematically. And this is the problem of targeting Cas9 cutting to amplified regions. Amplified regions are very common in cancer cells. Oftentimes, regions that contain an oncogene, for example, could be amplified. And in this case, what Wang et al. Noticed was that they looked at different cell lines, including K562 cell lines and KBM7 cell lines, and they looked at viability of cells with knockdown. This is actually coming from a genome-wide screen. So here you see all of these guide RNAs where they target along this chromosome. And they noticed that for K562 cells, there was this whole window of guide RNAs targeting this neighborhood on the chromosome that had strong growth defects in K562 cells, but not in another cell line. And it turns out that this is a region that is amplified in K562 cells. It is around the BCR-ABL fusion gene, which is actually driving that cancer cell. Interestingly, it's not only the guide RNAs targeting the BCR-ABL directly, but anything in that amplicon that are very toxic selectively in the K562 cells, whether or not they even lie on a gene. So the idea is that it could be that targeting amplified regions and just making a large number of cuts per se is non-specifically toxic and leads to dropout of those guide RNAs out of the population, but it doesn't actually reflect an on-target toxicity of the intended target gene of those guide RNAs. And again, you could say, well, that's an opportunity to learn about amplified regions, although that information often you can get in other ways. The problem, the potential caveat is that now it becomes very hard to distinguish within the amplified regions what are the driver genes, what are the interesting genes, which is actually a central question in cancer functional genomics. So that's an important caveat of these cutting approaches. Having said all that, they have been successfully applied in several areas. And a little later on in the talk, we'll look systematically at comparing some of the pluses and minuses of different approaches. So now I'm going to switch from talking to using CRISPR cutting in mammalian cells and using that for multiplex screens to using the technologies that Luke introduced, CRISPR-I and CRISPR-A, where a catalytically dead version of Cas9 is targeted to around the region of a transcription start site of an endogenous gene and used to recruit transcriptional repressors like the CRAB domain or transcriptional activators like in the case of the SUNTAG system, a whole array of transcriptional VP64 activators. And so this was really a big team effort of a team of folks in the Weizmann lab to turn that into a genome-wide screening platform, and really Luke and Max did all of the hard work here, and Max will be joining us tomorrow for the bioinformatics session as well. Basically, in this first paper, what we did is to derive rules for guide RNA activity in CRISPR-I and CRISPR-A settings in mammalian cells and conduct first genome-wide screens to see what we can learn using these technologies. So, okay. Based on the previous RNAi work, we had a pretty good idea of genes that would get strong phenotypes in pooled screens. In this case, we used actually a screen for sensitivity and resistance to a plant toxin called ricin. The phenotype doesn't matter. What matters is we knew certain genes, when we knock them down, we get a very strong sensitization or resistance to ricin. VPS54 is one example. And what we did, and really Max Holbeck drove the bioinformatics analysis on this, was to very densely blanket the area around the transcription start sites of these known hit genes with guide RNA and conduct a pool screen to quantify the phenotype of all of these. So we knew the expected phenotype based on the RNAi would be, in this case, for example, that we would get a positive phenotype here. So up on the y-axis are the guide RNAs that enrich in the population, and we knew that, and so we could use this to find out which guide RNAs or have a proxy for the guide RNAs that worked well in this setting. And one thing that's very visually visible here, both for DCAS9 and DCAS9 CRAB, is that there's a hotspot right around the transcription start site where these guide RNAs are very active. If you go further out, up and downstream, you lose that activity pretty fast. This is shown raw data for one specific gene, VP54, and we can now look at all the genes that were targeted in this pilot library and average the data, again centering everything to the annotated transcription start site. And what you can see for CRISPR-I is that there's actually a very narrow window just downstream of the transcription start site where you get maximal activity. And as Luke alluded to, that is actually a good thing, because what it essentially means is that even though per se it's hard to maybe design a guide RNA that's 100% specific in the whole human genome, most areas in the human genome don't matter. The only place you really need to check whether this guide RNA could have off-target effects are these narrow windows around transcription start sites. And so this is a huge advantage for specificity. Compare that to the case of, say, shRNAs, where even having, say, 6 to 7 nucleotide match anywhere in an mRNA could give you an off-target effects. This is off the charts more specific, just based on first principles. And we'll get into some data that illustrate that in a minute as well. One of the other important things, though, that comes with this is the fact that in order to be able to design good guide RNAs, you need to have a pretty good knowledge of where the transcription start sites are. And it turns out that depending which annotation system you use, the annotations are sometimes not that accurate. It is also a caveat that there seem to be some genes that have more than one annotated transcription start sites. Whether it's used or not is the other question, but that's a potential concern. And it turns out that, that relying on experimental data that actually did genome-wide experimental determination of transcription start site using, for example, a method called CAGE by the Phantom Consortium, these experimental start sites are quite reliable. And so far in our hands, we haven't noticed a lot of differences in transcription start site usage between different cell lines, I think. And so you know, I think this is still a young technology, so it is a potential caveat. But fortunately, so far, it seems that that this the annotated transcription start site is leading us in the right direction to have guide RNAs that work in a lot of different cells. Yes?
With CRISPR-A, are you ever changing the transcription start site, or can you activate a cryptic site? Because you're saying that I think with CRISPR-I, are you saying that you don't need to worry about this region outside of the known PSS?
That's a great question. So the question was, how about CRISPR-A? Can you also activate cryptic transcription start sites? So, so this is actually the equivalent figure for CRISPR-A. In fact, it's the same example gene, VPS54, which has the opposite phenotype when it's activated. So here, the optimal peak is slightly shifted. It's actually better to target a little upstream of the transcription start site. We think that this is because targeting this whole thing downstream the transcription start site, even independent of the CRAB domain, is obviously somewhat inhibiting transcription. And so to get the full benefit of activating transcription, you want to get out of the way of RNA polymerase. And that's probably why targeting upstream here is better. But as you can see, it's not like you would target it somewhere else and then have a strong activity. So you do have to have the normal context of the transcription start site, probably in terms of the cis elements. And another determinant is nucleosome positioning. So what's not so available in this paper, but what Max later on showed in a nice collaboration with the Tijan lab, is that it really matters to target these DCAS9 constructs to an area that's not occupied by the nucleosome.
Your point about the specificity is well taken. It's definitely nice that there's a narrower window to consider. As one maybe minor exception to that, how much do you think the effects of things we haven't really explained that well, like long non-coding RNAs, microRNAs, things that might control other parts of a pathway mimic phenotype if we disrupted them with off-target activity? Could you say anything about
Yes.
speculate will be the impact of those? And of course, it would be nice if I knew myself what percentage of the genome is estimated to be long encoding.
Absolutely. That's a great question. I think we're on safer ground talking about protein coding transcripts that are, I think, more of a consensus set. Once you start to look into non-coding RNAs, it seems that there's, first of all, any kind of database has a very large number of them because somebody at some point cloned something or saw something in one example. There seems to be some more, cell type and developmental specificity with respect to their expression. Obviously, the nice thing is, again, relying on experimental data sets that in an unbiased way just map where you see the relevant patterns, either nascent transcript, 5' ends, originating, or histone methylation patterns that are more characteristic of start sites and regulatory regions can guide you there as well. But at the same time, because I think with non-coding transcripts, there could be more context specificity. I think you're right that there could be many more transcripts, and the more context and cell types and so on we look at, the more of these transcripts we might find. And so you're absolutely correct in that that is a potential caveat. And I think I'll get to later in the talk for how to validate hits that come from a screen to exclude those kinds of false positives. So this is on-target specificity. Obviously, the flip side of the coin is off-target, or what does it take to be specific. And similarly to the data we saw yesterday already for cutting, our pilot library also included designed mismatches to the genomic target that had a phenotype, and so this is normalized. So the full phenotype of the perfectly matched guide RNA observed in this pooled screen would be 1, and then normalized to that, you can see how different mutations in different regions of the guide RNA, marked as gray squares here, ablate that activity. And so as you can see, it is definitely dependent on where you mutate. The closer to the 3' end and the PAM you are, the more dramatic the effect is. It's also dependent on guide RNA, so definitely mirroring some of the overall patterns we saw for cutting experiments. As soon as you combine several mutations, pretty soon you start to really lose a lot of the activity. And so Max, in his latest design, is incorporating these rules to select guide RNAs that would really have minimal expected off-target effects elsewhere in the genome based on these rules. All right, so the proof of the pudding is in the eating. How did the two compare? So I don't have the time today to take you through all the technical validation and all the data that backs up of this. As I said, this is published, but I think high-level summary is that based on our characterization, CRISPR-I compared to RNAi really has much reduced off-target effects. Nicely, it is also inducible and reversible. That data was already shown by Luke. You can, for example, have DCAS9 CRAB under doxycycline control. Important compared to some of these caveats I showed you for CRISPR cutting screen, it is non-toxic, so we can really, with a non-targeting RNA, maintain cells for a very long time, expressing DCAS9 CRAB, for example, even in a mixed population, and we maintain that stable ratio over time. So there's no selection against cells expressing the CRISPR-I machinery. One of the other nice things that Luke already showed in his talk was that CRISPR-I can be used to effectively target non-coding RNAs, which is not trivial with CRISPR cutting, because clearly, as we said before, CRISPR cutting would rely either on getting a frame shift, but there is no frame in non-coding RNAs, or knowing which regions are crucial for function, where a small indel might really disrupt function. But again, for these long non-coding RNAs, generally we have no clue. So this would be very hard to do in other ways. So Luke already showed you, based on an RNA-seq experiment, the really exquisite specificity in one example of targeting with CRISPR-I. Here we have a different experiment where we really base our observations based on the growth phenotypes of genes. So on the X and Y axis here, we see growth phenotypes of K562 human cells with different guide RNAs. Each dot corresponds to one guide RNA, and a phenotype of 0 corresponds to wild type. And actually, there's a very, very high density of dots right around here. So a lot of these genes don't have growth phenotypes. But then you see this tail here of really highly reproducible genes that had a growth defect, indicated by the negative phenotypes all along here. And yes, there's some scatter, which, as I said, any experimental method has noise. But one thing that is really quite nice is that not only did our library have negative controls that were designed to be non-targeting, and all of them essentially are really tightly clustered around no phenotype, the gray dots here. But we also had, you could always argue, well, maybe there was something weird about these negative controls. I don't trust that they truly represent off-targets or whatever. So one thing that we could do is look at the Y chromosomal genes. K562 cells are female, so they don't have a Y chromosome. And all of the green dots, again, are actually tightly clustered around here. Or olfactory genes, which are present in K562 cells but not expressed. And again, none of them had phenotypes. And so this kind of plot would look very, very different for RNAi, because even if you had shRNAs designed to target the Y chromosome or designed to target olfactory genes, as a general rule, they would have so many off-targets that some of them would hit genes impacting on growth, and you will see them have a phenotype as an off-target. So we were very happy with this. It seems to be also quite sensitive in that if we look at genes encoding subunits, for example, of the proteasome, the ribosome, or involved in DNA replication, which we would expect to be essential for growth. And so we expect those guide RNAs targeting these components to drop out over time, over the population. Here, this is a plot by the so-called GC method that we'll talk about tomorrow at the workshop. But you can basically see that the vast majority of these components clusters very close to the maximum phenotype you can get. So we get a pretty good coverage. And for example, for some of these ribosomal genes that don't seem to fall into that pattern, that don't seem to have such a strong phenotype, it turns out those were typically the ones that had a closely related homolog as well expressed. So potentially there was some redundancy. All right, so this has all been talking about CRISPR-I and what we can learn from loss of function. So one of the unique opportunities we have with CRISPR-A is to actually also look at gain of function phenotypes by switching on or overactivating gene expression. I showed you one example for a gene where that phenotype was exactly flipped, this VPS54 gene, and that was true for a few other genes as well from our original screen. But how about a cancer growth screen? And so here on the y-axis, you see gene phenotypes, on the x-axis, you see gene phenotypes from our CRISPR-I loss of function genes. 0 meaning, again, wild type, and negative phenotypes means that the guide RNA led to less growth or cell death, net cell death. And on the y-axis, you have CRISPR-A gain of function. And again, as expected, a lot of genes are kind of around 0, 0. A lot of genes don't have phenotypes. But then the genes that did have strong phenotypes are actually basically orthogonal. So you see different genes along here. And as I showed on the previous graph, the CRISPR-I phenotypes that were negative often represent genes that have housekeeping functions in the cell. Just important to keep it going. You see way fewer genes down here for CRISPR-A. So those are genes that are negatively impacting cells when the gene is overexpressed. So this is much sparser, but some genes have very strong phenotype, and they actually fall into very specific classes. They're tumor suppressor genes. That makes sense. So if you think of tumor suppressors, they're kind of the brakes on cell growth in cancer cells, and when you overexpress them, these cancer cells grow less. So that was true here for some of them. Or they were developmental transcription factors, so maybe driving these cells down a differentiation pathway, and that also interfered with growth, and some centrosomal genes. I'm going to go over this in the interest of time, but basically, integrating loss of function and gain of function data, you can even puzzle together some pathway that have negative regulatory information. So you really learn additional and complementary things from CRISPR-A and CRISPR-I. Mm-hmm.
And this type of screen has been performed with many different ways in the history. Have you discovered anything novel from this CRISPR-A and CRISPR-I screen?
I can show you a few examples that I'm now switching to a little bit more case studies where we definitely found some completely new things. I think these were things that were known in principle. This specific example here, that was known in principle, but obviously, as you know, it's always interesting to know which of these pathways are relevant and survival critical in a given cell line, right, for a given cancer cell. So I don't think that specific experiment has been done in K562. So if you will, we learned something new. But I'll show you some examples where we really learned even more fundamental biology.
Have any of those screens been done with CRISPR-I, CRISPR-A, and side by side with CRISPR cutting and RNA?
Yes.
That comparison?
There's been a few papers. A few papers came out recently, and.
Could you just say the question you were answering?
Oh yes, the question was, have any of these screens been done side by side using different CRISPR-based technologies and maybe RNAi to really directly side by side compare technologies in a setting? They have been done. There has been a paper from René Bernard's lab recently, and there has been a couple of papers from Mike Basik's lab recently. I decided not to put them in because my feeling and our own feeling from actually having done dozens and dozens and dozens of these screens is that we don't think necessarily that the lessons from these side-by-side screens were the last word, I would say. And the reasons are twofold. One of them was, I think, that all of these technologies are still actively developing, I think. In some cases, for example, the Bernard's paper has not necessarily actually used the state-of-the-art version of CRISPR-I, and then they came to the conclusion that it didn't perform that well, but they hadn't actually used the state-of-the-art implementation of the technology. So I think it's hard to make these conclusions. The Basic Lab paper had the conclusion between RNAi and CRISPR cutting that you actually get complementary information. I think that is true to some degree. You could imagine, and I'll get into that in a little bit, that CRISPR cutting can give you complete knockout phenotypes. So in some cells, you might get a complete 100% loss of function, versus RNAi or CRISPR-I give you some degree of knockdown. We can get very good knockdown, greater than 99% in some cases, but there's some protein left. And if you had a protein that really needs to be completely gone to see a phenotype, then you could only pick it up by a CRISPR knockout maybe. But again, having said that, in our own hands, actually, we get much better agreement between RNAi, for example, and CRISPR-I screens. So I think sometimes when people don't get good agreement, you also don't know, is that just like experimental variation? If they had done 2 different RNAi screens, would they also have found very different things, right? So I'm not citing these papers because I think it's still developing, and I think they're not the last word. But yes, people have done that, and I can send you the references if you want to look at these papers. Here's another example that really, I think, nicely illustrates the type of cell biology, for example, that we can learn, and shows how starting from one phenotype, in this case resistant to a specific bacterial toxin, we can actually pick up a lot about general cell biology in a quite, I think, impressive way. In this paper, we used a toxin, and we used this partially because we had made good experiences with toxins for technology development. These bacterial AB toxins, like diphtheria, cholera, and ricin that I mentioned earlier, are proteinaceous. They bind to cell surfaces. They're endocytosed. They often traffic in interesting ways through the cell and eventually target an essential gene function, often the ribosome or translation. And because they interact with so many host factors, they're a great tool to find a lot of genetic modifiers and learn cell biology. In this case, we used actually an artificial construct made by Hidde Plu's lab, and one of the motivations was that a lot of it was known, the biology, so we use this as a validation tool. And the technology that I mentioned very early on, this insertion mutagenesis and haploid cell line, had been applied to this exact toxin from that lab. So we kind of had a benchmark how that method compared. This toxin binds to a specific ganglioside in the plasma membrane, is endocytosed, traffics retrogradely to the endoplasmic reticulum where the subunits dissociate, and the catalytic A subunit traffics through to the cytosol. Presumably, it gets across the ER membrane through a channel that normally is used to extract misfolded proteins from the ER in a process called ER-associated degradation, and for degradation by the proteasome. But this toxin is not degraded. It actually has a catalytic activity. It's an ADP ribosylase, and it targets one very specific residue in a translation elongation factor, EF2. And that residue is post-translationally modified. It's a diphthemide. You already see that it's named, obviously, after the fact that it's targeted by diphtheria toxin. So this diphthemide residue is targeted, ADP ribosylated, which blocks protein synthesis and leads to apoptosis. We essentially did a screen, CRISPR-I and CRISPR-A screens, where we treated cells with this toxin at half lethal concentrations. We did this in several pulses, and then we simply asked what are genes that, when either knocked down or overexpressed, make cells either more or less sensitive to that toxin. And this is what we found. Here first the CRISPR-I data. Each of the dark red and blue dots represents a gene that was among the top 50 most sensitizing or top 50 most protective hits when knocked down. These light colors fill in pathways and protein complexes. And so what did we find? We found a ton of the very strong hits along this trafficking pathway, which was already known. So that was not a surprise. Interestingly, transport up to the ER, generally, when you knock down these factors, it's protective to cells. And we showed biochemically that knocking them down means that the toxin gets stuck in the membrane fraction, never makes it to the cytosol. As soon as you cross the ER membrane using some of these ARAT factors, the rest of the ARAT pathway now actually has the opposite phenotype. It gets sensitizing when knocked down. And that makes sense because the ARAT pathway normally leads to degradation, so presumably part of the toxin is degraded. When you knock down this degradative part in the cytosol, you in fact increase amounts of toxin in sensitized cells. Then we picked up essentially the whole diphthomide biosynthesis pathway. So that's also quite impressive because, as we said, diphthomide is the special residue in a translation elongation factor that's targeted by the toxin, and it seems like the cells are better off when they don't put the diphthomide on, but are no longer a target for the toxin. The translation apparatus itself is sensitizing when knocked down, something we find a lot. And then we also find the biosynthetic pathway for the cell surface receptor of the toxin, the specific ganglioside. And again, in a very nice way, we see that the enzymes that lead up to synthesis of the specific GM1A ganglioside are protective when knocked down, presumably because that means there's less cell surface receptor. The first enzyme in the pathway leading away from this metabolite is actually sensitizing. So presumably, if you can't change this into something else, you also have more receptor and you are more vulnerable to the toxin. So just by looking at this kind of color pattern here, you might have guessed what the cell surface receptor exactly was if you didn't already know. And in fact, we helped to specify it with this data. And the remaining top hits were probably pleiotropic. They had to do with gene expression. We do believe they were real because they were very tightly clustered and very few protein-protein complexes in the cell. And then these are the remaining ones in the top 50 that were all among the weaker hits down the top 50 list, and that are not part of these categories, some of them uncharacterized genes. They might be false positive, they might be new biology. We didn't follow up in this context. But I think the take-home message is that I think we found the screen was very sensitive and very specific as well, and actually I think much more comprehensive than the earlier haploid mutagenesis screen that found out the genes marked with little stars here. So we recovered essentially all of those, but many more to fill in the pathways as well. What do we learn from CRISPR-A in addition? Just focusing here on this ganglioside pathway, for a lot of the key enzymes, the CRISPR-A gene activation phenotype shown as the half circle here on the right versus the CRISPR-I phenotype is the half circle on the left. They often had mirror phenotypes, so in that sense they didn't teach us new things. But actually one thing you can see is that a parallel pathway that starts from the same precursor lactosyl ceramide here, we picked that up only by CRISPR-A. Presumably it's a pathway that's not very active in those cells, but when we artificially activate it using CRISPR-A, we can divert some precursor into this branch away from the branch that actually makes the receptor and thereby makes cells actually more resistant. Okay, so the obvious question, and Lin, you asked that already, is how do we compare between these different approaches? How are they different? What if you want to design an experiment, what should you choose? I don't think there's a perfect answer. This is from a nice review here by Fan Zhang's lab that shows one important difference that I alluded to. Down here we have in panel B, it's actually in some sense easier to explain. We have RNAi or CRISPR-I systems, and what we're looking at is gene expression level for an unmodified cells here in orange compared to a controlled cells where that gene is not present or expressed at all. And so basically the idea is that in our experimental system we knock down the gene. It might not go all the way down to non-expressed levels, but what is important is that the whole population behaves consistently. And this pretty much mirrors the experimental data that Luke in fact showed you just an hour ago. The difference with Cas9 cutting is the following. Again, we have the orange, which is the unperturbed cells. The dotted would be a cell that doesn't have that gene. But then with Cas9 nuclease, we don't get the whole population behaving as one. We can get different outcomes. We can get some outcomes of cutting that, in fact, don't change the level of functional protein at all. Maybe something was introduced that just deleted an amino acid that was irrelevant for function. We get some things that in diploid cells might be one allele effectively inactivated, the other one still there. And then we get some that are completely off. And what the ratio of these are, I think it depends. Some of these nice technologies, nice new technologies that Dana mentioned might be able to take advantage of the knowledge of microhomology to in fact bias guide RNAs towards getting more efficient frame shifts. So again, it's hard to compare the technologies because all of them are still improving and developing. But this is the fundamental difference, I think. You can get complete knockout with CRISPR cutting, but you generally don't get it in all the cells, so you get a heterogeneous mix. Versus with RNAi or CRISPR-I, you don't get complete knockout, you can get pretty good knockdown. But the good thing is the cells behave more or less consistently in the whole population. So to compare a few other things, one of the advantages of RNAi is obviously it's a one-component system because you only introduce shRNA or siRNA, the rest of the machinery is already in cells. So for some systems, it could still be the method of choice, and we have certainly continued to develop best state-of-the-art RNAi libraries for those cases. Versus for CRISPR-I or CRISPR nuclease, we need at least two components. Inactivation, knockdown versus complete null but limited efficacy, as shown on the previous slide. RNAi and CRISPR-I are reversible. This is actually very nice for if you wanted to manipulate gene function in a time-dependent way, in a time-controlled way, versus CRISPR nuclease is not reversible. In terms of specificity, RNAi is obviously notorious for off-targets, as we said, which is a problem in genome-wide screen that you can overcome with complex libraries, but that comes at a cost. CRISPR nuclease is somewhat specific, and I think it's still an improving system in terms of specificity, but there is a concern. Versus CRISPR-I, really based on our data, seems to be the most specific one right now. In terms of toxicity, RNAi can in fact be toxic if it's not done right. If you have very, very high levels of siRNAs, you can overwhelm the RNAi machinery. But generally, a system where you express things from a Pol2 promoter inside a microRNA context doesn't seem to be terribly toxic at all, not in our hands or in terms of what people publish. CRISPR-I, again, we tested, is not toxic, versus CRISPR-N does cause DNA damage. It causes a DNA damage response. And as we saw based on these papers, that can actually definitely impact your results in a pooled screen. And targeting non-coding RNAs is difficult with RNAi and CRISPR nuclease, but it's definitely feasible, and Luke showed the data with CRISPR-I. So this is just kind of an overview of these three choices that I think are probably the top three choices for people who want to do loss of function genetics at this point. I have a more detailed list here going also through all of the other approaches I showed on my first couple of slides, but in the interest of time, I'm going to skip that. You'll have the slides ready for download. But I think the short answer is, while some of these things are, interesting, they are not. Random mutagenesis could really generate interesting alleles that have new functions that hit something very interesting about the genes, and that's a huge advantage. It comes at the disadvantage of practicality. It's much harder to map things that you randomly introduce as opposed to this targeted approach that we discussed here. And some of these other approaches that are targeted, like zinc fingers, it's not that feasible to do a genome-wide screen with zinc fingers if you have to engineer a new protein for each gene. All right, so I'm going to skip over these things. Okay, one thing now that we talked about tools is to talk more about strategy. And by that I mean, how do you even design a screen? What are the kind of things you can look at, and what do you need to be careful about? And so I want to first introduce this distinction between a screen and a selection. We've been talking about screens, and most people call everything a screen, so I'll probably stick to that. But strictly speaking, if you want to make the distinction, you could say a screen is one where each phenotype is individually evaluated, and that could be a case where you, for example, have a 96 well plate and you do microscopy on each well, or you do some other measurements, and versus a selection strategy which used a pooled approach, which is all of the CRISPR-based technologies I mentioned to you are pooled approaches to this point. It means that the cells or organisms with the phenotype of interest are somehow pulled out of the population, either based on their survival or resistance to a stress, or by other methods of physical separation that I'll show. And these have their pros and cons. The screen requires arrayed reagents to perturb gene functions, and for example, I think it's only a matter of time that we have arrayed reagents for example, CRISPR cutting and probably also CRISPR screens. We're not there yet. The pooled approach enabled the whole technology to take off within months because of this arrayed oligonucleotide synthesis that enables you to really very rapidly generate genome-wide libraries. Screens require a high throughput platform to actually determine the phenotype. So not every lab has access to a high content microscopy platform that can go through thousands of wells, versus a pooled selection scheme is essentially accessible to everybody who has a tissue culture setup. The big advantage of screens are that you can screen for very complex phenotypes. You could screen for non-cell autonomous phenotypes that involve interesting interactions between different cell types. You can screen for things in a time-dependent way. So versus for selection-based experiment, only certain phenotypes are suitable, and I'll come back to that in parallel. Certainly because in a screen you look at things one thing at a time, that really limits the scale much more than the parallel evaluation of phenotypes, which is essentially just a space issue but not a time issue, because you're evaluating everything in parallel in a screen. Here in a screen, you know what genetic perturbation you're intending to apply in each well, because you have your arrayed libraries of, for example, guide RNAs. In the selection-based experiment, you use deep sequencing to really interpret this connection between genotypes and phenotypes, which actually means you need genotype-phenotype coupling. So that means you need, in this case, to integrate guide RNAs, for example, into the genome stably, so you can actually recover what was the genotype in these cells that we selected out here. And essentially, that means you can only screen cell-autonomous phenotypes, right? You couldn't answer a question such as, okay, I have tumor cells and stromal cells, and I'm interested in tumor cell survival, but I want to find out what are all the genes in stromal cells that matter for this. That doesn't work. You have to perturb the gene in the same cell where you read out the cell-autonomous phenotype. Versus here, the genotype-phenotype coupling in a screen is actually given through physical compartmentalization. You have your well where the cells are, where the reagents are, and that gives you the coupling that you need. Technically, I think, and especially also in the light of the arrayed screens that have been done, for example, these siRNA screens for HIV factors that I showed initially were arrayed screens, and they're really notorious for things like batch and edge effects, right? Because these things are looked at sequentially. There are some clever ways to try to correct for these things, but I think the nice thing about these pooled screens can be that they're really done in a homogeneous environment. All the cells are in the same environment, and providing a better internal control. Okay, as I said, selection experiments have certain prerequisites for you to be able to do one. As I said, genotype-phenotype coupling is important, so we need to make lasting changes to the genome. This phenotype needs to be cell-autonomous, and what is such a phenotype? We can definitely look at growth and survival, and there were some examples already just looking at things impacting growth and survival, say, of cancer cells. You can do that in the presence of a specific selective pressure. The example of the bacterial toxin was such a selective pressure, and as you saw, you can pull out all sorts of cell biology from applying an interesting selective pressure. but there's a whole other classes of questions that you can't couple to cell survival or cell growth, but that you can solve by physical separation of cells. And examples for that would be anything where you can build a fluorescence-based reporter for your phenotype of interest. And I show some recent and unpublished examples for that later on. So, because then you can use FACS sorting to physically separate out cells that have the phenotype of interest. And again, in those separated population, then look which genes are over and underrepresented in those. And in theory, you can also do things like an affinity-based cell purification, right? So if you're interested in what all determines expression of a certain cell surface marker. For example, in the context of cell differentiation, in theory, you could also physically pull them out with magnetic beads. Or you could even look at phenotypes like cell migration, right? So do cells respond and chemotax properly? Is that a way to physically separate them? And can I find out which genes, when knocked down, prevent that from happening because those cells won't migrate? So those are the things. So, in the remaining time today, I will cover one important type of application that has many possible outputs, but the basic strategy is always the same, and it's looking at gene-drug interactions in this following scheme that looks familiar from before. We create our libraries of cells in which each cell has a gene knockdown or overexpressed using shRNA or guide RNAs for CRISPR cutting or CRISPR-I. And then we split it in half and grow one half by itself, and the other half we treat with a drug. And generally our strategy is to treat with the drug around the EC50 so that we kill about half the cells, but then we can let them recover and repeat that pulse several times, 3 or 4 times, in order to apply the relevant amount of selective pressure. And we'll talk much more about the quantitative aspects of this tomorrow. But what do we get out of this? One thing, and I show you a few examples for that has been very powerful, is to use this to identify the direct target or the mechanism of action of a drug. This is actually a big question that both academic science, but also certainly pharmaceutical industries and biotechs are interested in. A lot of new drugs come out of phenotypic screens. So much like the arrayed siRNA screens that you could do in wells, you can also put drugs in the different wells, and you have a cell-based readout for a, say, cell-based disease model that you want to see. Again, the simplest will be a drug that kills a cancer cell. And but oftentimes, it's actually not trivial to then, once you have a compound, identify what is the mechanism, what in the cell was the compound hitting. There are biochemical approaches. You can synthesize cross-linkable analogs of the compounds and then physically try to pull out proteins it interacts with. That has several caveats, sometimes putting a cross-linker in there actually changes what the compound does. Physical interaction doesn't mean that it's a functionally relevant interaction. And while it has helped in some cases, there are many cases where that approach doesn't work. So as I show you, we can use genetics to identify genes that either where the knockdown sensitizes to the target or desensitizes to the compound, and that can lead us to really identifying what the relevant target in the cell is, which is hugely important to know if you want to further develop the compound for use in patients. Another area that is obviously also talked about a lot is precision medicine. So the idea that patients are not a homogeneous population. Different patients are different, and ideally you want to know what are the things that are predictive in a patient for response to a given drug. Ideally you want to identify what are the biomarkers that I need to look up to decide, do I give patient drug A, B, or C, or maybe none of them will work. Also, especially in the context of cancer, cancer cells develop drug resistance, and we'd ideally like to be able to predict all the possible routes to drug resistance, not by giving it to a patient and seeing them relapse, but predict it already in vitro before it first sees a patient, because in that case, maybe again, we could come up with a rational combination therapy or way to overcome that drug resistance. And lastly, the long-term solution probably to drug resistance in cancer is going to be combination therapy, just as it was in HIV therapy and with certain antibiotics. It's easy for evolving cells to become resistant to one single agent at a time, but it's much less likely, even just based on first principles, for a cell to be resistant against maybe three or so different compounds. But we don't want to maybe pick them at random. We'd ideally like to predict them. So let me show you a few applications where we successfully use genetic screens to do all of this. A first example was a collaboration with Mike Cleary's lab at Stanford, who had found a compound that was very active against a very high-risk form of leukemia that's found in children. And again, they didn't know what the target was, so we did an shRNA screen at the time, some time ago. This was done in experimental replicates, so 0 means wild type, and negative numbers means sensitization to the compound when knocked down. Each black dot is a gene, and you can see most genes are clustering around wild type, a little bit of correlation here, but there was one single gene that really stood out completely. This gene encodes an enzyme called NAMPT, which is involved in NAD+ biosynthesis, and our prediction based on this was that this would really be the direct target and the relevant target of that drug. And there was actually, interestingly, a way to prove that, because the cellular need for NAMPT can be overcome by supplying cells with nicotinic acid. So here's the result. In red, this was the kill curve with the compound on these cells, very steep kill curve. When we supply nicotinic acid, cells become completely resistant. So that nicely supported biochemically that this gene was in fact the relevant target.
This wouldn't work with a knockout, right? Because you would kill the cells.
Yes, that's a great question. So certainly one of the things that Luke already pointed out, and that I haven't made strongly as a point, is that a complete knockout for essential genes wouldn't work, right? And especially in this context where you're targeting clearly something that is essential to cells, you may not find it with a CRISPR knockout. Having said that, in diploid cells, maybe knocking out the cells that have one knocked out, you could pick up a signal from them, but yeah.
Where do you think CRISPR stands in this continuum of, knocking out where essential genes are not going to, especially essential drug targets are not going to come up, versus shRNA, where you know that you're not really knocking out everything. So do you think CRISPR is would pick this up or?
Yes, and we have our first CRISPR screens as well that have been performing well at identifying drug targets and identifying essential genes. So I think one of the nice things of these pooled libraries is that we clearly target each gene with more than one guide RNA. And if you think about it, even though it's not a design feature a priori, it so works out that some are more active than others. So in some sense, you get an allelic series of different knockdown strengths. And so we've been definitely able to pick up those signatures. Yeah. Mm-hmm. Here's a more complex phenotype because, again, it seems maybe facile to always talk about cancers and killing cells, but this is a much more complicated example. This was a collaboration with Peter Walter's lab, who has done a compound screen to inhibit a stress response in the cells called the integrated stress response. There is a fluorescent reporter for this response. I won't go into details, but basically, if you activate stress in the cells and you monitor the levels of this fluorescent reporter, you can suppress the reporter in a dose-dependent way using this compound. The compound had very interesting property. For example, it can enhance memory in mice, but the cellular mechanism and the actual target of this compound was unknown at the time. So I generated a reporter cell line for the stress response, and we had a scheme where we induce stress either by itself with a small molecule that induces endoplasmic reticulum, ER stress, or in the presence of half maximal concentration of the compound, leading to either more or less activation of this fluorescent reporter. And then we fact sorted cells that had either the lowest third or the highest third of reporter levels in both cases. And then we asked what gene knockdowns are enriched in those populations, what are the phenotypes. And this is a complex experiment. Just to summarize and say that on this axis we have the screen without compound. Here we have the screen with compound. Things that affect reporter levels with and without compound are along the diagonal, but we have a few genes that were very specifically interfering with the reporter in response to only in response to drugs. So those would be along the axis here. And the two most prominent things right along the axis were actually encoding subunits of the same protein complex. When knocked down, the reporter was higher. So essentially the compound wasn't active at repressing it, and it turned out that this compound was in fact directly binding to and dimerizing this protein complex and activating it. So that's why in this case, knockdown actually desensitized to the compound. So that is a nice example where we picked up something that didn't impact growth at all, but having this more complicated phenotype of interfering with the stress activation of a reporter, we were able to figure out the compound. An example where we fully took advantage of CRISPR-I, CRISPR-A is a new compound developed by a company called Cleave Biosciences that was designed to target an important AAA ATPase in the cell called P97VCP. But again, while you can show in biochemically that it's binding and inhibiting this protein in a biochemical assay, it's not that obvious to prove that this is a relevant target in cells. Typically what people do is they take a whole cell line panel and determine how the compound, the IC50 of the compound across these cell lines, and then they can correlate it with gene expression. In this case, expression of the direct target gene P97VCP. And they saw some amount of correlation, but it's actually very low, right? It is significant because it was a large number of cells, but this could have been, to call this a causal is quite a stretch. So instead what we did is we generated a whole series of guide RNAs that would either knock down or overexpress P97 in an isogenic background. And what you could see then is actually a very, very tight correlation with sensitivity to the compound. So this shows the power of what I was mentioning really early on, that say using natural cancer cell lines, you're always underdetermined, right? There's so many differences between any of these two cancer cell lines. Yes, VCP expression is one different, but so many other things are different. With CRISPR-IA, we can fine-tune expression levels of one single gene in an otherwise isogenic background, and in this case got a very clean answer that levels of VCP expression in both directions from wild type really controlled sensitivity to the compound. All right, so I think what I'll do is, well, I should briefly go over this. So these were all examples that characterize the mechanism of a compound. This is an example, a published example, where we really go more in the direction of precision medicine using an inhibitor of the proteasome that is clinically used in a type of cancer called multiple myeloma. There are compounds like bortezomib and carfilzomib that have actually been successful in a clinic. It's a little surprising because all cells need the proteasome, so what determines that therapeutic window? Also, unfortunately, patients all become resistant. What are mechanisms of resistance? And can we identify pathways that are synthetic lethal? And without going into any details today, we were able to really find things that control sensitivity in either direction, and then work with clinicians to show that expression levels of those genes also were predictive of patient response. So that in our cell-based model, we were in fact able to identify predictive biomarkers. We are also able to predict good ideas and bad ideas for combination therapies. For example, what we predicted and what had been published before is that knockdown of HSP90 chaperones would synergize with proteasome inhibitors. So we get a negative sensitization to carfilzomib. But what we predicted is that knockdown of mTOR, which is actually also considered as a drug target in cancer, would actually be a bad idea because it protects cells from this knockdown. So you have two drugs that individually are killing cancer cells, but we predicted that in combination, they would actually desensitize things to carfilzomib, and we could show that by pre-treating with this inhibitor, we would actually slightly desensitize cells to carfilzomib, even though by itself it's toxic. So again, we can also start to predict combination therapies that way. I think at this point I'm going to stop talking about examples of screens in biology, and tomorrow we go more into the nitty-gritty of analysis, and I take questions as well, but during that time I'm going to put up this slide. So for the computer workshop tomorrow, you will have to download two things. You can do it tomorrow at the workshop, but I'm also putting it up in case you want to do it already. So Max Hallbeck, who will be here tomorrow, has put together a folder on his GitHub account with all the scripts you'll need. And so if you go to github.com, mhallbeck screen processing, this is the screen you see, and there's a little green button where you can click on it and say download zip and download it to your laptop already. That has all the scripts you need. And then These scripts are based on Python 2.7, so that doesn't mean anything to some of you, I think. For others, there's Python 2 and Python 3, so this is Python 2 base, 2.7. So if you have that on your computer already, great. For some of the functionality where we're going to interact with the data in a visual way, you are not just running Python 2.7 from the terminal is not ideal. So what we recommend for this and other applications too, is that you can download an environment such as this. This is a program called Canopy Express. You can download it for free from this website, nthought.com. You go to this website, you click on products, you click on download Canopy, and there's a free option. You can download it, and that will enable you to run these scripts in an interactive way where you can visualize data. And so that's what we recommend for tomorrow. So I'll leave this up for a few minutes while I take questions.
Going back to the off-target effects, how much is known about the on and off about the CRISPR complex? And is there a rational way by looking at the guide sequence to find the optimum KD that is required, and then go back into the genome again and say, well, these guides are going to be specific because they have an X amount of KD versus the other guide RNA?
Right. Yeah, I think that's a great question, and I think partially what Dana covered yesterday, the same question applies to obviously both cutting and to CRISPR-IA. I think clearly the idea is that some of the off-target effects and the non-specificity comes from the fact that in some sense you have more affinity than you need, and so by weakening strategically some of these interactions you could gain more specificity, including weakening interactions on the protein side, right? And so these more engineered versions of Cas9 for increased specificity will probably help, and certainly I would predict that they would also be good for doing these CRISPR nuclease screens. For the CRISPR-IA system, I think it's something we would have to evaluate systematically, and we have the tools to do that to see if some of these mutants improve specificity. But the short answer is there was really very, very almost no detectable off-target activity even in genome-wide screens with this, that we are sure this is an off-target effect. And so it's hard, what are we even optimizing for? And I think, again, the main reason for that is probably that most off-targets of the CRISPR-I system are going to be irrelevant unless they're really in that narrow window in a transcription start site. Mm-hmm.
Because one thing we always worry about are the SNPs and the promoters, and the SNPs maybe
yeah. Yeah, absolutely. I mean, one region where clearly that matters is the PAM, right? So SNPs around there, if you can use a system where you can take advantage of that, or the most other sensitive reason for most systems is close to the PAM, right? So.
Other question is, what's known about these guide RNA? Can they interact with some other machinery? Can they interact with microRNA machinery? Is there some kind of evidence or a pull-down experiment that suggests that they are exclusively getting with the Cas9 and nothing else?
That's a great system. Certainly, I think in our hands, the guide alone control is something we obviously worry about with any new system, and we haven't really seen phenotypes for that before, right? I mean, correct me, Luke, if you've seen other things.
Some of the RNA-seq I showed, like with express just guides on their own, or Cas9 on its own.
Because there's a new report which suggests that ergonaut complex can be complexed with guide RNA and break the DNA, which is independent of PAM sequences. So I'm just wondering if some kind of interference is occurring in these guide RNAs as well.
It's a good question, and if you have seen that paper, I'd be very interested in that people have directly shown that as a substrate. Because clearly, as soon as you have then again short interference in RNA, there could be some off targets. Again, based on what we observe, I don't think this is a major pathway in our hands, but that would be important to look into. Yeah. Mm-hmm.
So my question is related to the consistency between multiple guides of the same gene. So I know that the problem, especially with small libraries, is that the lack of consistency, because especially if you're using like libraries with a low number of sgRNAs per gene, there's a lack of consistency. So my question is, I know that in your libraries you have at least like 10 sgRNAs per gene, right?
Yeah, the version 2 libraries that are currently under review that Max Hollbeck designed have 5, and it seems that you can really, in terms of detecting hit genes, there's no big loss to go from 10 to 5 with the newest design, and that is because quite a good fraction of them tend to be active. You're not going to get always the exact same phenotype because the knockdown level will still vary, and it's still something we can't predict that well, but. I think the biggest problem comes from completely inactive reagents, right? And then if you have a very small library, like a 3 per gene library, having 1 or even 2 inactives will really throw you off, right?
Right. So my question is, when using like 5 guides per gene, on average, can you tell like how much of them for true candidates will be like functional or active? So I can rephrase the question if you like. Did you identify true candidates with only few sgRNAs showing consistency?
So what that means is, it depends a little bit what your approach to calling hit genes is, which is what we'll talk about in depth tomorrow. And you can have an approach where you have certain requirements to call something a hit gene, right? A lot of traditional approaches use, especially from the RNAi field, use hard cutoffs. They say we need to see at least 2 shRNAs or 2 siRNAs that have at least whatever 4-fold effect or something like that, right? We generally don't use that approach. What we use instead is a Mann-Whitney rank sum test to look at the entire set of negative controls and the entire set of guide RNAs and say, is there a significant shift? It takes into account both the magnitude of the shifts as well as how many shift by how much. So it's a slightly softer and more integrative metric. But there are different approaches. Yeah, And I think some of what you're saying will very highly depend on the overall noise level in your screen, right? Because the observed phenotype in your raw data will be very strongly dependent from how noisy the screen was overall. And so that's one of the reasons that in our libraries we always include lots of negative controls, because they should all have no phenotype. So depending how the observed phenotypes for those spread, that gives you a pretty good idea of the noise levels in your screen already, right? So a lot of the specific answers to your question will also depend on how well was the screen designed and carried out. What's the overall noise?
More questions for Martin? No?
I'll be around tomorrow all day, so you can ask me more then as well.